Next, we're going to have Hans van Volkoven from Radboud come and present. He has very similar information to some other things that have been talking about regarding, how are we going to further express EHMT1 and different methodologies that we could use. So, Hans, when you're ready, I will turn it over to you.
Yes, thank you, Jeff. I'll try to share my screen. Do you hear me?
Hear you very well.
Oh, thank you very much. And now you should see my screen as well. Is that correct?
Yes, sir. It just loaded. We are good to go.
Yeah.
Yes, we can see it. It's good.
Okay. Yeah. Well, thank you, and thank you for inviting me to talk at this meeting. Yeah, I've been involved in Kleefstra syndrome genetics for a longer time. Let me see how I move around here. And actually, in our lab at Radboud University, Netherlands, Nijmegen, we have been studying all sorts of neurodevelopmental disorders for decades, to be honest. And of course, well, the entire genetic field has exploded by the application of next-generation sequencing tools. By now, and I just made a calculation yesterday, more than 1,600 different genes have been identified that if they carry a mutation, give rise to some kind of neurodevelopmental disorder. And well, one of these is Kleefstra syndrome caused by EHMT1. Of note, the vast majority of the dominant type of disorders, like Kleefstra syndrome, involve haploinsufficiency, and I'll come back to that later, what that actually means exactly. Just a short history on the syndrome, how we got involved with it. Well, the syndrome, of course, as you are all well aware, is named after my colleague, Tjitske Kleefstra. And actually, when she was a PhD student in the group, we were very much excited in the identification of genetic causes of mainly X-linked, X-chromosomal type of intellectual disability. And our research on Kleefstra started with the patient shown in this photograph, who carried the translocation between the X chromosome and chromosome 9. And initially, we had been very excited about the breakpoint, chromosomal breakpoint on the X chromosome, as we thought that the gene on that chromosome would be causative for the phenotype in this girl. But then soon after, we realized that in fact it was not the breakpoint on the X chromosome, but it was the breakpoint on chromosome 9 that was more relevant, because the phenotype of this particular patient resembled that of the 9q subtelomeric deletion syndrome. Now, after that, it became apparent that it was not just the entire part of the tip of chromosome 9 that was important, but just the EHMT1 gene that carried causative mutations, loss of function variants in the gene. Now, after that, we started to study the EHMT gene, as well as other genes that are involved in chromatin type of processes, chromatin-related processes, in different European collaborative projects. And yeah, actually that learned us a lot. And well, I'll just give you an overview of a few things that we have done in the past years after the identification of EHMT1 as the causative genes. But it mainly started with a human phenotype and the clinical information as a basis, and of course the patient material, to model the disease and to learn more about the mechanisms of the neuropathological phenotype. And for that, we used mouse models, and we learned a lot about mouse behavioral and processes that went eerie in the mouse model that we had studied. The same for Drosophila models that we had used throughout for studying again the behavioral phenotype, the processes involved, and also the extensive repertoire of epigenetic mechanisms that was involved. But lately, the last, let's say 5 to 7 years, we have been taking advantage of the study of human-derived material, and in fact, in patient-derived induced pluripotent stem cells and neural lineages derived thereof. And you have heard at this meeting already a lot more about that from my colleague Neil Nadev-Kasri. Of course, we are very, very keen in getting insight into mechanisms of disease, but ultimately, the purpose also of the work that we are doing is to try to take advantage of the new knowledge that is being generated to find new targets for therapy and to find tools and to create tools that could be used for preclinical testing of possible interventions that might be applied to patients with Kleefstra syndrome. But the strategy that is commonly used nowadays is starting off with patient material, fibroblasts, skin cells, or blood cells that we use most frequently to generate induced pluripotent stem cells. And this is done from control individuals, but it's also done from patients with specific mutations in our gene of interest, the EHMT1 gene, of course, in the case of Kleefstra syndrome. And besides those patient cells and control cells, we also use CRISPR-Cas9 editing technologies to introduce mutations in a control cell line, so to mimic mutations that have been found in individuals with Kleefstra syndrome, or vice versa, to rescue mutations in IPSCs that have been derived from a patient cell. So that gives us access to what's called isogenic lines to do comparative studies. Now, from these IPSCs, we use different protocols to differentiate those into different neural lineages. Most frequently, we are using glutamatergic cortical neurons that are derived from the IPSCs. So this is what we use most often in our studies. But at will, we can, or with other, let's say, transcription factors activation, we can also generate inhibitory parvalbumic neurons, and also with other protocols we can generate astrocytes. We can use these differentiated cells in isolation, in cultures, in monocultures, or in cultures with that are mixed, that have mixed types of neurons. And mixed means the different neural cell types that can be used, but also making different combinations of mutant and wild type cells. And that allows us to get insight into certain processes and neurophysiological processes that are disrupted in Kleefstra syndrome. Now, what we typically do is culture the differentiated neurons on so-called microelectrode arrays. Neil Nadev-Kasri has been talking about it. And when seeded on those microelectrode arrays, after a while of differentiations, these cells start to form connections and neural networks that have intimate contacts with one another, and intensively and spontaneously communicate with one another with minute electrical signals that appear as either spikes or at trains of activity that's being called, they're called bursts. And we can analyze the patterns of spiking and bursting, especially the network bursts that indicate the connectivity in the culture system to derive the physiological properties of the cells. Now, when these properties are visualized in graphs that are indicated here, then what we see is a typical patterns of spikes, the small spikes that you see here, and bursts. This is a train of spikes that are appearing very fast after one another. And when such bursts are seen in all electrodes in the culture disk, we call them network bursts. And this means that there is a connectivity has occurred in the cells that allow the spontaneous collective bursting activity of the neurons in that culture. Now, this is the typical pattern that one can appreciate when control neurons are analyzed on a microelectrode arrays. If we do the same experiment, but then with neurons that are generated from patients with Kleefstra syndrome and with mutations in EHMT1, we also see a pattern of spiking and bursting, but you can easily appreciate that this pattern is totally different from the pattern that we see in controls. So the pattern of bursts in Kleefstra syndrome is much more irregular. The duration of the burst is much longer, and the frequency of the burst is in general also reduced. So they have a lower frequency than in the control situation, whereas the spiking activity is still very high or relatively high as compared to the control. So this all indicates that the network organization is reduced for Kleefstra syndrome derived neurons. Now, this is a pattern that is very typical and let's say stereotypic for Kleefstra syndrome, and we use that abnormal pattern of on microelectrode arrays for several purposes. We use this sometimes, or we can use it as a diagnostic tool to have an interpretation of variants of unknown significance that ever so often are identified in our diagnostic laboratories by whole genome sequencing of patients with an intellectual disability phenotype. And sometimes a variant of unknown significance is identified in a gene of interest, for example, EHMT1, where we cannot decide just by the sequence change whether it's causing the disease. Well, the interpretation of a pattern that I just showed on the previous slide could assist in the assignment of causality of such VUS. What we mostly use these IPSC-derived neurons for is studying mechanisms of disease, and I'm sure that Neil Nadev Kasri has already told you a lot about it. And I will just summarize one of those results and then further concentrate on how we envisage to use also these iPSC-derived neurons for testing therapeutic targets and preclinical testing. Now, what Neil and his group has been doing is to analyze the neuronal network of Kleefstra syndrome neurons. And as I mentioned in the previous slides, what is being observed is that the pattern is very different from the pattern of control neurons in three aspects. The duration of the bursts is longer, network burst frequency is lower, and the network organization is longer. Now, a pattern like this can also be measured at the single cell level, and it's indicative of the disruption of the activity of certain receptors. And in this case, the pattern that we had observed also at the single cell level is indicative of an increased activity of one of the major synaptic receptors, the NMDA receptor, as indicated in this slide. Now, this suggested that something is wrong with the NMDA receptor. There's higher expression or higher activity. And when we looked at the components, the NMDA receptor is a multi-protein complex. When we looked at the different subunits of the receptor, we saw that in the neurons that were analyzed on the microelectrode array, there was an increased expression at the RNA level of one of these subunits of the NMDA receptor, the GRIN1 subunit. Moreover, there was not only an increased expression of this subunit, there was also a noted decreased methylation at the position that was used by EHMT1 for its methylation, this lysine 9 of histone 3. So due to the hypothesis is that due to the reduced activity of EHMT1 in patients, there is decreased methylation at some target genes, one of them being GRIN1 as a subunit of the NMDA receptor. There's more GRIN1 subunit, which leads to an increased activity of NMDR, and that leads to the abnormal pattern that we observed on the microelectrode arrays. Now, this was supported, this hypothesis, by the fact that we were able to use an NMDA receptor antagonist, as indicated by the name over here, and when we apply that antagonist, we could see a normalization of the microelectrode array pattern on the system. So a normalization towards the pattern that's also seen in control neurons. Now, this is an example of how the system can be used to identify mechanisms of disease, but also to test components that have the ability to normalize an abnormal neurobiological pattern and the microelectrode arrays. And this is a scheme that you see here in this slide is a scheme that shows the heterogeneity of the genetics in neurodevelopmental disorders. And what we very often observe in these neurodevelopmental disorders, that there's many genes, but ultimately the disruption of those genes gives rise to common disruptions in biochemical processes, in cell function, in morphological features of parts of the brain, for example, and ultimately also in common behaviors and phenotypes. And of course, this is also the case for EHMT1, where we see that, and also other chromatin modifiers, for example, who would affect the same biochemical processes, functions, and phenotypes. Now, for the NMDA receptor antagonist, it's mainly an antagonist that would have an action at the level of cell function, morphology, and perhaps ultimately also some symptoms that are manifest in the disease. Same can be said for other small molecules, for example, olanzapine, that's currently being used in a clinical trial by Kleefstra and colleagues. Also, this is typically a small molecule that would act on large-scale, let's say, cellular functions and downstream mechanisms that are relevant to the disorder. However, these do not act on the root cause of the disease, and that's the defect at the genetic level. So the deficiency of EHMT1 protein. And this is where we are interested in. We are very keen to get more to use the haploinsufficiency observed in Kleefstra syndrome as a tool for intervention by upregulating the expression of EHMT1, just like we have heard in the previous presentation by Dr. Ulitsky. So EHMT1, I don't think I need to explain, is on chromosome 9 with the DNA structure indicated over here and the protein structure indicated over here with all its, with some of the mutations that we know by now. But important to highlight is that the mutations that are typically associated with Kleefstra syndrome are either deletions, entire or partial deletions of the gene, or single nucleotide variants that diminish or abolish the function of the EHMT protein. So efficiently, there is a mutation of one copy of the gene that leads to inactivity, and only one remaining copy is still active. Now, this is highlighted here. Again, a diagram of the normal situation with 2 copies of EHMT1 in a normal individual, leading to a certain level of transcripts at the RNA level, and that's being translated, giving rise to the adequate amount of EHMT1 protein. In the disease situation, there is haploinsufficiency, and 1 copy is affected. That leads either to reduced transcription, a 50% reduction of transcripts, or to reduced, well, let's say, functional protein because of a loss of function mutation in the protein. The net result is that the activity of EHMT1 is decreased by 50% in patient cells. Now, what is still there is 1 normal copy in the patients with Kleefstra syndrome, and this is immediately, well, the target for intervention is how to upregulate the normal levels of EHMT transcript or protein. There are several ways to do that, and some of them have already been highlighted in the previous presentation. But it's a very, well, critical question, because it's not as easy as it seems. The problem is that you need to achieve this in the cells that you want to target in the nervous system. In our case, it's mainly in the nervous system that we like to do our targeting. Also critical is to reach the right amount of upregulation of EHMT1. The Kleefstra syndrome is caused by a reduction of 50% of EHMT1 protein, but we know that also an overexpression of the gene and an over amount of EHMT1 protein is also pathological. So this is something that one also wants to avoid. And if you think about regulatory mechanisms, the question is also, how long will your upregulation that you want to achieve, how long will it last? And of course, most of all, is your intervention going to be safe? Now, a lot of potential strategies exist, and I've listed well, a variety of them in the slide over here. So we know about gene therapy, antisense RNAs, microRNAs, modulating of splicing, acting and modulating cis-regulatory elements. CRISPR technology is a possibility with using modified Cas9 molecules that can have an effect on transcription levels. And also enhancement of translation with certain molecules, synapse is a possibility. Now, all of these strategies have their pros and cons, and I can quickly give you a glance of a few of these. Well, gene therapy is one option, but this is typically achieved by viral delivery of a gene, of a normal gene copy. But it's not as easy as it seems, and the difficulty and the problem for EHMT1 is mainly in the accurate dosing. So it's going to be relatively difficult, we estimate, to have the accurate dosage and the right amount of EHMT1 expression by using gene therapies. Sign-up is another technology. I'm not going into the detail of that, but sign-up is a technique that takes advantage of the RNA molecules for EHMT1 that are still present and boost the translation of the residing RNA molecules into EHMT1 protein. Now, this is done by a special vector, a sign-up vector, that allows the increased translation of the residual RNA molecules into EHMT1 protein. Also in this case, well, the delivery is not the biggest problem, but also here the accurate dosage is the biggest challenge that one can face in this technique. Still, it's an approach that is being pursued and is useful to pursue, I would say. So we, at the moment for EHMT1, but also for other genes, are very much interested in using antisense RNA molecules, just like what we have heard in the previous presentation. And we like to apply to EHMT1. Now this, the use of antisense RNA molecules and long non-coding RNAs have been well introduced already in the previous presentation. So I'm not going to reiterate that, just to say that, that it takes advantage of non-coding RNAs that are very abundant in the genome and that have, well, in many cases, a regulatory role on the expression of genes and mainly genes that are in the vicinity of those non-coding RNAs in a normal situation. And the idea is to exploit this activity, so exploit the normal activity of existing non-coding RNAs to modulate the expression of the gene of interest. Now, examples have been given. So this strategy is used for a number of other disorders. Angelman syndrome, Dravet syndrome, and Chaser have been mentioned. For EHMT1, this is now just in progress, and we heard about RDDC1 antisense molecule that's also indicated here in this slide. That can be used as a possible antisense target for intervention. And this is indeed just much the same as what we have heard in the previous presentation, what we are now in the progress of investigating to see how these non-coding RNA molecules are potentially involved in the regulation of EHMT1 expression, because this has yet to be firmly established. And second, if they are, how can we modulate the expression of those antisense RNA molecules to also modulate the expression and upregulate the expression of EHMT1? Well, besides these antisense non-coding RNAs, there's also other RNA molecules, and I just highlighted microRNAs that are also potential regulators of EHMT1. Now, the advantages of antisense RNAs as a vehicle to modulate expression of RNA targets has been well established. Also, the tools for delivery have been well established, including delivery in the central nervous system. So this has been applied for in preclinical testing in animal models, but also ongoing in clinical trials in humans. And of course, well, we have yet to see also for those other disorders the efficacy and the duration of any effect that might be brought about by the use of antisense RNA molecules. So at the moment, what we are now doing is to correlate the expression of the antisense RNA molecules that are shown and identified with the expression of EHMT1 itself. And we are doing that in different cell types, but mainly, of course, cell types, neuronal cell types derived from Kleefstra syndrome patients, as well as control lines that we have. Second, we like to develop, like in the previous presentation, antisense oligonucleotides that potentially have an effect on the expression of these antisense RNAs, and by that also of EHMT1 expression. And then, of course, if that has been established, we like to investigate in our iPSC-derived neurons what the neurophysiological consequences are. So can we, by applying antisense oligonucleotides, normalize the electrophysiological patterns, the neural network activity that can be measured on microelectrode arrays? And well, this is work ongoing. There's still a lot to do. We love, of course, to collaborate, and I hear mentioning by Dr. Ulitsky that he is in touch with my colleagues at Radboud UMC. So it will be great to work together on this and see if we can identify something, an antisense RNA molecule that has efficacy on EHMT1 expression. Now, this is the work of many, and well, the PIs on the project, the main ones I've already mentioned, Neil Nadev Kasri and Tjitske Kleefstra. I also should mention Dirk Schubert, who also has a pivotal role in all the work that I've been presenting. So thank you very much, and I'll be happy to take questions.